Let us study a new chapter, Introduction to Microbiology. What is meant by microbiology? Microbiology is a branch of science in which we are going to study about different types of microorganisms. Now, microorganisms speciality, those organisms which we cannot be able to see with our naked eyes. For seeing them, we have to use microscope. Now, different types of microorganisms are bacteria, fungi, viruses, protozoans or protozoa, parasites, then algae. These are the different types of microorganisms. Now, there are two branches of microbiology. One is known as pure microbiology. Pure microbiology. The other one is known as applied microbiology. These two are the branches of microbiology. In pure microbiology, we will study about different types of microorganisms and their structure, then we can say shape, even size, then we can say that in them which type of DNA is present. That means we can say that their genetic materials, then we can say that their organelles. All these things we are going to study in pure microbiology. At the same time, in applied microbiology, we are going to study their applications. Applications, it's a meaning uses, or we can say as uses. So here pure microbiology, we are going to study about different types of microorganisms, then their genetic materials, cell organelles, then types of microorganisms, about their DNA, etc. At the same time, about applied microbiology. In applied microbiology, we are going to study their uses. In this chapter, we are going to study more about their uses. We can say that microorganisms are used in various fields, like we can say agriculture, then we can say that food production, then we can say that medicine production or medical field, we can say. Then we can say that chemicals production, then maintenance of garbage maintenance, then we can say that pollution control. So here we can say that we can use different types of microorganisms for, we can say different types of food production, we can say, then dairy products production, then in agricultural field, garbage maintenance, then pollution control, different types of chemicals production, then different types of medicines production, even different types of protein production. Then we can say that different types of nutrients production. Here all we can say that with the help of microorganisms, a process is happening. That process we can say is fermentation process. Fermentation process. With the help of fermentation process, different types of microorganisms are helping for different types of uses, we can say, for the production of different materials. There are useful bacteria as well as harmful bacteria, or we can say useful microorganisms as well as harmful microorganisms. So in case of food production, agricultural field, even garbage maintenance, we can say that again pollution control. Here all means we are using microorganisms for our benefits. So we can say that they are useful microorganisms. Now we will study in detail about microorganisms and for which use, which type of microorganisms we are using it. An important field in microbiology is industrial microbiology. Industrial microbiology, we can say that it has very much use in commercial purpose. We people are making so many things. We can say, for example, bread, then wine, then we can say that medicines. These all things means large production of such materials are taking place in the industries and selling and getting benefit from that. So large quantity of production of anything is happening for getting benefit. We can say that that is industrial production. So here microorganisms we are using for even industrial purposes. So as a result of it, we are calling it as industrial microbiology. What are the main features of industrial microbiology? With the help of fermentation process, various microorganisms are useful for making different products like bread, wine, cheese, and raw materials for raw materials for medicines, enzymes. Enzymes means proteins, chemicals, nutrients, etc. Then again, so here we can say that first feature we can say because of or various process of or with the help of fermentation process we can be able to make various products like wine, cheese, bread, even raw materials for making medicines, chemicals, then we can say nutrients, enzymes, etc. Other thing, even we can use microorganisms for maintaining garbage, or we can say garbage maintenance. garbage maintenance as well as pollution control. Let us study the definition of applied microbiology. What is applied microbiology? Applied microbiology is a branch of biology in which study of enzymes related to prokaryotes and eukaryotic microbes, proteins, applied genetics, molecular biology, etc. is performed. Once again we will study what is applied microbiology. Applied microbiology is a branch of biology in which study of enzymes related to prokaryotes and eukaryotic microbes, proteins, applied genetics, molecular biology, etc. is performed. Let us study the basic process of the production of yogurt, cream as well as cheese with the help of a flow chart. Such a chart, this is known as a flow chart. Here again flow chart is there. Then our study work will be very easy. Here now first what we have to do, we have to take required quantity of milk. Then we have to do pasteurization process. What is meant by pasteurization? We have to boil it. We have to boil the milk. When we boil the milk, the disease germs or the harmful germs which are present in the milk, they will die. Then we have to add or we have to ferment this boiled milk. So first we have to boil the milk. Afterwards we have to keep some time for becoming lukewarm milk. Once lukewarm milk, that means just we have to keep our finger, then our finger won't get boiled, means burned. Such a condition, warmth will be there. In that we have to add a kind of bacteria. That bacteria is known as lactobacilli. This, when we add lactobacilli inside the lukewarm milk, its fermentation process will begin. For completing the fermentation process, one overnight we have to keep it. One night full we have to keep it. As a result of it, here the conversion of lactose sugar, the sugar which is present in the milk, that is known as lactose sugar. That lactose sugar will convert into lactic acid. Then once lactic acid formation will take place, the milk converts to a thick proteinaceous substance. We can say that that is the yogurt, or we can say that a sour taste will be there, such a substance will form. So that process is known as a thick substance is forming from the milk as a result of formation of acid, lactic acid. That process is known as coagulation of milk. So this is the basic process for the production of yogurt, cream as well as cheese. Now in our home, when we people are making for domestic purpose, we can say curd we are making at home. So hearing the word of yogurt as well as curd, in our mind confusion will be there. Yogurt as well as curd is same. But curd means we are making at home by using lemon juice or vinegar or little curd. We have to add in the lukewarm milk. So we can say that curd that we people are, or buttermilk also we can say, curd or buttermilk, that we are making at home, either by using lemon juice or by using vinegar or by using little curd in the, or by adding little curd in the milk. These three things we have to, out of these three things, any one little quantity we have to add in the milk. Then if you are keeping overnight, or we can say one night full, the milk turns to curd or buttermilk. At the same time, yogurt means that is a commercial product. For that we people are adding two types of bacteria inside the warm milk. As a result of it, curd like a product is forming. So that is known as a yogurt, but yogurt is a commercial product. At the same time, curd or buttermilk that we are making at home. I can show you a simple product of curd or buttermilk which I made at home. Here you can see that water like thing you can see. This is known as a whey. This whey, if want we can utilize, or if we don't want we can throw it out. Whey, W-H-E-Y. Whey means the water which is removing out after the coagulation of the milk. So the thick substance will separate, then water content which is present in the milk, that will separate. The water content which is separating from the milk after becoming curd, buttermilk or yogurt or cheese, that is known as whey. Now we will study commercial preparation of yogurt. Here again we have to boil the milk. After that we have to cool it for some time. That means no need of cooling it fully till it become lukewarm. Till the time we have to cool it. Afterwards we have to add two types of bacteria. First bacteria is known as Streptococcus thermophilus. Streptococcus thermophilus. Then Lactobacillus delbrueckii. Lactobacillus delbrueckii. We can say that equal quantity of both of them. Suppose if you are taking Streptococcus thermophilus 1 spoon, then Lactobacillus delbrueckii also we have to take 1 spoon. That means 1:1 proportion we have to take both the bacteria. Then we have to keep it for fermentation process. As a result of fermentation, there will be the formation of lactic acid. Once lactic acid formation will be there, we can find that coagulation process will take place in the milk. That means a thick substance will form, that is known as proteins gel out. Then that will be a thick substance, we can say that will be a thick yogurt. So here what is given? Proteins gel out, giving a thick or dense consistency to the yogurt. Yogurt means a curd like thing. Curd only we can say. Curd, this is commercially preparing one. The curd which I shown you, this is a homemade one. Here what I done, I taken warm milk. In that I added some quantity of curd inside. Instead of curd, milk, warm milk we can take. In that 1 lemon juice we can pour it, or 1 spoon or 2 spoons of vinegar we can add it in the milk. Then 1 night full if you are keeping, that milk transforms to curd. So there we can say that conversion, when milk transforms to curd, you can find that thick substance is forming. Milk turns to now a thick substance. So we can say a dense consistency is occurring, thick substance is occurring. Coagulation of milk is happening. Thickness is occurring to the protein which is present in the milk. So that is known as here proteins gel out saying. Or here we can say coagulation of milk. So this thing means domestic purpose we are making, or we can say homemade one. At the same time, for commercial purpose, that means a large quantity of curd making, that process is known as or that commercial process of making yogurt. There means a large quantity of yogurt is making. For that they are not adding curd in the milk, they are adding two types of bacteria in it. One bacteria is known as Streptococcus thermophilus, other bacteria is known as Lactobacillus delbrueckii. As a result of it, fermentation process will begin. One whole night if we will keep, there will be the formation of lactic acid. Lactic acid is having sour taste. Then as a result of the formation of lactic acid, the conversion of milk into a thick substance, we can say that proteins gel out. Then that yogurt will be having very dense or thick consistency. Now we are studying about butter. There are two types of butter. One is known as sweet cream butter. The other one is known as cultured butter. For making cultured butter, we have to use microbes or microorganisms. Now we will study how to make cheese commercially. Commercial production of cheese. For that we have to take milk, normal milk, no need of boiling the milk. But before making the cheese, we have to do the chemical as well as microbiological test. After that, we have to add inside the milk color as well as three types of bacteria. They are Lactobacillus lactis bacteria, Lactobacillus cremoris bacteria, Streptococcus thermophilus bacteria. All these bacteria we have to mix it. As a result of it, the milk turns to a sour substance. We can say curd like it will become. Then we have to slow by slow. That means as a result of adding all these things, the milk transforms to or turns to coagulated substance or a thick substance will become. Then that thick substance we have to separate and remove. Plus the milk, when it transforms to a thick substance, the water content how much is present in the milk, that will separate as this, almost like this. We can say that here water is separating, thick substance is separating. So that thick substance with the help of, this is means overnight we have to keep it. But at the same time, for making cheese, as and when we mix all these bacteria, the process will take place faster. Then coagulation process will take place. As a result of coagulation, a dense consistency will occur in the milk. That dense substance or that thick substance, with the help of a spoon we have to separate it. Then water particles of the milk will separate. The water content how much is present in the milk, that is known as whey. The thick substance we will separate it. After that, next step we have to do. So when we add these bacteria, milk become sour. Sour means its taste will be like curd. After that, thick substance we have to separate it. Water content of the milk we have to separate. The water content that we can separate it, its use is not coming. So that water's content we can call as whey. That thing we have to remove. Now the thick substance is there. That thick substance we have to separate it, and all the water content we have to separate it out from it. Then it will become a very thick substance. Remove all the water content from it, whey from it. Then a thick substance we will get it. Then that thick substance we have to put it in the warm water. Then we have to wash it. Again, all water content we have to press it and remove out. And like a dough, a thick substance, a ball-like substance will form. Then that process, that thick substance we are making it. Like a ball we are pressing it and removing all water content after washing. That process we can say as rubbing. Then that thick substance without water we can say, in that adding salt. So for what purpose may be adding salt? Salt is a preservative. So for keeping for long time they are adding salt in it. After that they are adding again some essential microorganisms. We can say some more bacteria they are adding it. And pigments means color they are adding. Again we can say flavors they are adding. Flavor means a smell getting substance they are adding. After that again they are pressing the cheese. Again they are pressing, any water content is there, that water content again should go out. For that again they are pressing it, making a ball like shape. How much water content is there, everything they are removing out step by step we can say. Later they are cutting that cheese into pieces and they are storing it or storing for ripening. That means we can say that if you are keeping more days, then it's that stretching ability. You may be knowing in the pizza they are putting cheese. Then as a result of it, you can find that pizza, when we are cutting and removing, thread like a thing is coming out. That is because of the help of cheese. So once cheese is getting heated, we can find that it is stretching. Thread like it is becoming. So how much more very good quality of cheese want to make it? For that we have to preserve it or we have to store it more days. Saying that minimum 3 week time if you are keeping it, that much good quality curd, this cheese will become. Or more, how much more you want, that much more days you can keep it. So more days if you are keeping, that much good quality of cheese can be made. So once again we will study. This we are making for commercial purpose, that means for making large quantity of cheese. What we people are doing it, or the industrial preparation of cheese, what people are doing it. First what they are doing, they are checking or they are testing the chemical as well as microbiological test of milk. For making cheese there is no necessity of boiling the milk. Now in that milk they are adding microorganism, that means a bacteria, different types of bacteria as well as color they are adding in it. Which type of bacteria they are using? Lactobacillus lactis, Lactobacillus cremoris, Streptococcus thermophilus. These 3 types of bacteria they are adding it. As a result of it, milk becomes a curd like, sour taste one. After that, once such a thick substance, coagulation process will happen, then the water content which is separating from the milk, that is known as whey, that they are removing. Other thick substance, coagulated substance we can say, thick substance, they are removing. Afterwards they are pressing it, removing all water content from it. Then they are washing with warm water. After that again they are pressing it, removing all the water content from it. Then they are pressing a ball like thing they are making after removing all water content, that is known as a rubbing. In that they are adding salt. As a result of adding salt, the salt itself is a preservative. So we can say that shelf life of cheese will increase. After that they are adding some essential microbes means microorganisms, then some color as well as some smell getting substance, that is flavor they are adding. After that they are once again any water content is there present, any water content is there inside the cheese that they want to remove. For that again they are doing the pressing work. After that they are cutting the cheese into pieces. After that they are storing it for ripening, that means for becoming a very good cheese. They are keeping it for long time in the we can say freezer or fridge. What are probiotics? Probiotics are food materials, but in them there will be active microorganisms or microbes. In probiotics, even milk products are coming or including. We can say that yogurt is an example of probiotic. Similarly, kefir, some milk drinks are there. We can say that fermented milk products, they are also the examples of probiotics. Then pickles, what we are having at home. There are different types of pickles. In them all, active bacteria is present. Because of the help of active bacteria, our alimentary canal means mouth, esophagus, stomach, then small intestine, large intestine, then anus, rectum, anus. So that means the whole we can see from mouth till the anus. A tube is there. In that these microbes will grow. As a result of it, our digestion process will take place in a proper way. These microbes grow in colonies. Colonies means in group. And as a result of these microorganisms, these microorganisms are very useful to us. They maintain balance in the digestive system. As a result of it, if they increase the digestion process and they increase our resistance power. If there is any harmful microorganisms in our body, to them they will kill, they will remove. And in case if we take any antibiotics, the useful bacteria or useful microbes which are present in our body, they will become inactive. But when we consume probiotics, in that active microorganisms are there or microbes are there. So as a result of intake of probiotics, our inactive microbes will become active. Now in probiotics, which all types of microbes are present? Let us see that. Lactobacillus, acidophilus, Lactobacillus casei, Bifidobacterium, etc. Etc. means a number of. So few examples given here. More than that, different types of microbes are present inside the probiotics. Probiotics are food materials. Examples we will study. Which all are the, or what are the examples of probiotics? We can say that we studied even milk products are coming in it. Here you see yogurt, kefir. Kefir is a drink, milk drink we can say, fermented milk drink. So yogurt, kefir, then sauerkraut, that is pickle of cabbage. With cabbage also people are making pickle. That cabbage also an example of probiotic. In that active microorganism will be there. Then dark chocolate, then one special type of soup, its name miso soup, then different types of pickles, oil, then corn soup, then artificial sweeteners, then microalgae. For microalgae, different examples given, that is different types of seafood like spirulina, chlorella, blue-green algae, etc. So all these food items are the examples of probiotic. If we consume probiotics, that means the food material which contain active, useful bacteria or microbe we can say. If we consume such food items, our digestive system will be function in a very good condition. There will be the balancing or there will be or they will maintain a balance in the digestive system. As a result of it, the number of useful microbes will increase and harmful microbes will decrease. For harmful microbe, there is one example, that is Clostridium. Clostridium bacteria is a harmful bacteria. Clostridium bacteria is an example of harmful bacteria. How to prepare bread commercially? You all may be knowing bread is very fluffy, spongy. How it is making? That is making with the help of carbohydrate. Carbohydrate means flour, wheat flour. Then in that baker's yeast they are adding, salt adding, then water adding. Only 4 ingredients are there. Plus something else, little oil, such and such things also may be adding. So for making bread commercial purpose, that means for selling purpose, how they are making it, that only shown with the help of this flow chart. First here baker's yeast given. Baker's yeast, that means it is a microorganism. Its name is Saccharomyces cerevisiae. Saccharomyces cerevisiae. This is baker's yeast. It's a scientific name, Saccharomyces cerevisiae. Then water, salt, flour, wheat flour, even some other necessary materials like oil we can say. Then all these things mixing together, we have to make a dough with it. Then we have to keep it overnight. That means 1 night full we have to keep it. As a result of it, fermentation process will take place. The carbohydrate or sugar that converts to carbon dioxide as well as ethanol. Now, as a result of formation of carbon dioxide, when we bake the bread become fluffy as well as spongy. When we cut a bread or when we remove a bread piece, you can find that many holes inside. These are holes formed while baking. Carbon dioxide gas escaped out. So how much gas was present, that is gone away. As a result of it, their holes formed. So once again we will see how to make bread commercially. For that we have to use 4 to 5 ingredients. Baker's yeast, its scientific name is Saccharomyces cerevisiae. Then water, salt, flour, flour means wheat flour, then other necessary ingredients means oil, etc. Then we have to make the dough. For making chapati, how we are making dough, same way we have to make it. Then 1 night full we have to keep for fermentation. As a result of fermentation, the carbohydrate or the sugar content which is present inside the flour, that converts to carbon dioxide as well as ethanol. This carbon dioxide will come out when we bake the bread. As a result of it, we will get fluffy as well as spongy bread. Now, here we can say that because of the help of this baker's yeast, only the fermentation process is happening. Now, we will study what all ingredients are present inside the baker's yeast. This baker's yeast, we can say that compressed yeast. It will be available in the form of small granules, or even now almost powder like it is available. That is known as instant yeast. Now, which all ingredients? Baker's yeast or compressed yeast or instant yeast, containing 5 different ingredients are present. They are carbohydrates, fat, proteins, vitamins as well as minerals. As a result of it, with the help of baker's yeast, which all food materials we make, all those food materials will be nutritive, highly nutritive food materials. So when we eat such food items, it will be giving us good nutrients. No, there will be no harmful effect. Now, Chinese food, all are aware of Chinese food. In that Chinese food, 3 important ingredients they are adding. One is known as monosodium glutamate. Monosodium glutamate. That is its scientific name. The scientific name of what? Ajinomoto. Ajinomoto is the common name. Its scientific name is monosodium glutamate. Second thing, vinegar. Third thing, soya sauce. These 3 things are the main ingredient of the Chinese food. All these important ingredients of Chinese food are produced by microbial fermentation. So we can say that they are fermented food materials, which are monosodium glutamate or Ajinomoto, vinegar and soya sauce. These things are producing by microbial fermentation process. The word vinegar is very much familiar to you all because it is adding in different types of food materials like ketchup, chutneys, pickles and different types of sauce. Vinegar we can make in 2 ways, one chemically, the other one biologically. Now chemically, vinegar contains only 4% of acetic acid. Out of 100%, 4% of acetic acid and 96% of water. Biologically, we are making vinegar from ethanol. Now, how to make ethanol? Ethanol is making either by using fruit juices or maple syrup or sugar, molasses or starch of the roots. Now here, different types of fruit juices we can use, or maple syrup means it is getting from maple tree. From rubber tree, rubber milk how it is getting, same way from the maple tree its syrup is getting. From one tree 20 to 60 liters of syrup getting. Then they are heating it and concentrating it, making it a thick solution. Then sugar molasses means from the sugarcane juice they are refining the sugar. Afterwards, which waste product is forming, that is known as sugar molasses. Then starch of the roots, for example tapioca, that is the root of a plant, tapioca plant. Then its syrup, they are taking it, they are boiling it, then juice they are taking it. In that juice they are adding yeast. All these things mixing with the yeast. Here, scientific name of yeast is Saccharomyces cerevisiae. As then they are keeping it for few days, we can say that more than 10 days they have to keep it. As a result of it, they are getting ethanol. This ethanol they are mixing with the bacterial strains. Here 2 types of bacteria they are using. One is Acetobacter bacteria, other one Glucanobacter bacteria. As a result of it, the decomposition or degradation process of ethanol will take place. As a result of it, there will be the formation of acetic acid and some other byproducts will form. Then they are heating this mixture, that is only known as rarefaction, or we can say distillation process. As a result of it, vapor of acetic acid collects, then they cool it. Afterwards, they bleach it with the help of potassium ferrocyanide. Bleaching process means a cleaning process. Once after bleaching, they do the pasteurization process. Pasteurization means a heating process. Then they are adding little quantity of sulfur dioxide gas. As a result of it, we will get vinegar. Thank you.